Hey Lab Squad! Ever spent weeks optimizing a plasmid, swapped out promoters, add enhancers, only to stare at a Western blot that screams low expression? It's frustrating, right? What if I told you the problem might not be your promoter, but something way more subtle happening after transcription? Today, we're diving into the tissue-specific tRNA pool and why ignoring it is killing your protein. Let's get to it. So, you've heard of codon optimization, right? It's where you swap out codons in your gene for preferred ones in your host organism. The idea is that common codons lead to faster translation and therefore, more protein. It sounds great on paper. But here's the catch. When you just say optimize for human, most online tools use a general codon usage table for the entire human genome. It is based on an average, and as such, often hides the really important stuff. Your body isn't just one big blob of human cells. You've got brain, gut, bone cells, and many more. And guess what? Each one has a slightly different pool of tRNAs and crucially different amounts of those tRNAs. Imagine your ribosome, scooting along your mRNA, to pick up tRNA carrying an amino acid. If your target tissue has tons of a specific tRNA, the ribosome finds it instantly and the translation is fast, efficient, and the protein gets made. That's a preferred codon in that specific tissue. But what if your tissue only has a tiny amount of the tRNA needed for a certain codon? The ribosome hits that spot and stalls. This is ribosomal pausing. The protein is halfway made and starts to fold incorrectly. It aggregates, and the cell tags it for degradation. So you get less protein, or worse, useless protein. And here's the crucial point. Because of tissue-specific tRNA scarcity, the amount of protein you can make is capped by the rarest tRNA needed for your gene. Simply put, you can never make more protein than the amount of the rarest tRNA present in that specific cell type. Next time your expression fails, remember, it often comes down to the tRNA pool in your specific cell type. Optimizing your gene to fit the codon bias improves your yield, enhances mRNA stability, and even influences immunogenicity. And now for the tips. Tip number 1. Get tissue-specific codon tables. Don't use generic human optimization. If you're targeting liver cells, use a liver-specific codon bias table to match your gene to the specific codon bias. Check the TissueCo-Coput database for these specific tables. This free public database gives you the actual frequency data. Use it to create a custom codon table, ensuring your gene truly matches the tRNA capacity of your target tissue. If your target tissue is very unique, just build your custom table. Go to a public database and select the most highly expressed genes from that tissue. Retrieve a reasonable number of gene sequences, as FASTA file, and concatenarize them. Run their combined sequences through tools like CodonW or CA-ICL. This gives you the precise frequency data you need for the best optimization. Tip number 2. Watch out for rare codon clusters. While one rare codon is a small delay, a string of them is a disaster. These clusters cause serious ribosomal stalling, which ruins the protein and drastically cuts your yield. Be sure your optimization strategy actively removes these clusters. Tip number 3. Mind the GC content. Don't just optimize codons. Messing with the overall GC content can destabilize your mRNA. Tissues have unique optimal GC ranges, so true optimization means balancing codon usage with mRNA stability. Okay, so you have your custom table and optimized your gene for the perfect tRNA pool. But there's one last critical step. Tip number 4, the final quality check. Changing the sequence can accidentally create or destroy functional sites. Always screen your new sequence for 2 critical risks. CPG islands that can lead to disease, like cancer, and cryptic splicing sites that will break your RNA. Run your final sequence through a bioinformatics tool to make sure your perfect gene doesn't become a perfect disaster. Can you do this optimization manually? Yes, although it is time-consuming. But for large gene batches, you need the power of professional bioinformatics tools. Modern expression is deeply intertwined with computational science. Embrace it, and your lab work will thank you. By addressing the tRNA pool of your specific tissue, you move beyond simple gene optimization and tackle the fundamental physical process of translation, guaranteeing that your hard work on vector design actually pays off in high protein expression. Drop any questions in the comments below. Don't forget to like this video and subscribe to the Lab Survival Guide for more tips to make your lab life easier. But my cat also just reminded me, it's Sunday, how about some rest? See you next time, Lab Squad.